Uh, Yesterday's introduction of Plasmodium almost completed. So, uh, 4 marks question is there. Describe the structure of sporozoite. This is one of the stages in the life cycle of Plasmodium vivax. Sporozoite is an infective stage of malarial parasite to man. At this stage, we will get this disease. We will be infected with this stage only. After that, it will be developed into the trophozoite that feeds on hemoglobin, our hemoglobin, causing the malaria fever. So, this sporozoite is the infective stage of Plasmodium to man. That is the first point you have to write down. Infective stage to man. You say like that. It is infective stage to man, humans. This monograph on sporozoite described by the scientist Shorten Granham. Actually, Granham described, but it is given clearly described by monograph. Generally, monograph, you know what is monograph? Any particular organism, if it is taken and description starts, then it is called monograph. So, on sporozoite, this scientist Granham, Shorten Granham already it is referred in the case of pre-erythrocytic cycle. So at that time, the Granham separated from the short and described about the sporozoite, details of the sporozoite. So described by Granham, first scientist already previously in the introduction and history of malaria, I have mentioned the 5 scientists. First Charles Leveron, second Sir Patrick Manson, third Ronald Ross, and fourth Grassi, and fifth Golgi. Camillo Golgi described the life cycle in the erythrocytes of the human beings. Erythrocytic cycle was discovered by Camillo Golgi, who discovered the Golgi apparatus in the cell. The same scientist also described the life cycle of Plasmodium in our RBC. So, described by Granham, this is sickle shaped. You know, sickle is one of the agricultural tools with which we cut the grass. So, grass cutter is sickle. Sickle in Telugu we call it kodavali. So, in villages you will come to know about this because you are all from urban area. You may not know about hoe, this plow and all these names. So, like that this is also sickle. Spade means para. Sickle means kodavali. Hoe means buriga. Plow means nagali. Like that there are all agricultural tools. So, it resembles a sickle. Hence, it is called sickle shaped. And both ends are pointed. Both ends pointed. Like this, you can make it a short form of a sentence. Both ends pointed. And 15 microns in length. Length of sporozoite is 15 microns. Microns symbol, you know, this is. And width of the sporozoite is 1 micron. Width is 1 micron, length is 15 microns. This is about the size of the sporozoite. And it is centrally located nucleus is present. Centrally located nucleus, but not cartwheel-like nucleus. Cartwheel-like nucleus is a characteristic feature of the trophozoite of Entamoeba histolytica, but this is simply centrally located nucleus. And body is covered by pellicle. External covering of body is pellicle with microtubules. Pellicle with microtubules. In the pellicle, these microtubules also present. Because of these microtubules, it shows wriggling movements. Wriggling means like this. You see my forefinger, I am moving. So this is called wriggling movement. So shows the wriggling movements. Sporozoite shows the wriggling movement is a assertion. Reason is the pellicle of sporozoite contains microtubules. That is, so shows the wriggling movements. Kind of locomotion in sporozoite is wriggling movements in the blood circulation of humans. Generally, it shows that movement. In the anterior end, there is an apical cup. Apical cup present at the anterior end. at the anterior end. I will draw the diagram also. You can see this, all these structures you can see. Apical cup at the anterior end. Into the apical cup, the secretory organelles open. One pair of secretory organelles. Secretory organelles how many? One pair. In the bracket you write down one pair. Secretory organelles open into the apical cup. Open into apical cup. So that's why the sporozoa, otherwise called epicomplexa, already have given the classification of Plasmodium vivax. In this phylum Protozoa, subphylum epicomplexa we have mentioned yesterday. Epicomplexa also called sporozoa. Epicomplexa is given due to the presence of apical cup and along with secretory organelles. It looks like a complex in the anterior. That's why it is called epicomplexa. Now you know that the reason why it is so called epicomplexa. Now we can understand this. So apical cup at the anterior end, into which the secretory pair of secretory organelles open into the apical cup. These secretory organelles secrete the enzyme. Secretory organelles, enzyme secreted by secretory organelles is cytolysin. Organelles secrete the enzyme cytolysin. Previously in the entoma histolytica, that is histolysin. Histolysin dissolves the tissues, but cytolysin dissolves the cells. So secretory organelles secrete cytolysin, which helps in the entry of sporozoites into liver cells, which helps in the entry of penetration or entry of sporozoite into liver cells. Sporozoite first attack the liver cells. Into liver cells. Liver cells technical name is hepatocytes. Liver means hepato. So if you want technical term of the liver cells, you can write in the bracket hepatocytes. They are hepatocytes. You can put it in bracket. So that is the function of secretory organelles. For the entry of sporozoite into liver cells, these secretions are helpful. And in the cytoplasm, unknown function convoluted tubules also there. Function is not known. Unknown function convoluted tubules. Convoluted tubules of unknown function. You can also say like that. Present in cytoplasm of sporozoite. All these structures you can see in the diagram. And ER, Golgi apparatus, mitochondria, lysosomes are all present. Lysosomes, ribosomes, they are all present. Are all present in the sporozoite. So see the diagram now. With the help of diagram, if the diagram is asked or not, it is immaterial. But if it is asked or not, you have to draw the diagram. That is an additional impression. Okay. So describe the structure of the sporozoite with the help of the diagram. Like that also question may come, or it may not come that describe the structure of the sporozoite without asking diagram. Then also you have to draw the diagram. It is best. It is always better. So this is the sickle shaped sporozoite with apical cup like this, and centrally located nucleus is present. This is centrally located nucleus. All these are convoluted tubules. These are all many convoluted tubules. And ER and all these structures are present. And in the anterior end, a pair of secretory organelles are present which open. This is one secretory organelle and this is another secretory organelle which open into the apical cup. So now let us take the labeling of the sporozoite. So this is apical cup, secretory organelles. Just now I told you which secrete the enzyme cytolysin, and convoluted tubules, and this is nucleus, pellicle, with microtubules. This is labeling. Okay, that is sporozoite. Now, life cycle of Plasmodium. Let us take the life cycle of Plasmodium. This is the heading. Life cycle of Plasmodium takes place in two hosts. Hence, it is digenetic parasite. This is from this two essay questions are coming. Life cycle, it completes its life cycle in two hosts. So each host 1 8 marks question. Life cycle in man and life cycle in female Anopheles mosquito. So here life cycle of Plasmodium. This is digenetic parasite. Introduction. Digenetic cytozoic parasite with two hosts. Namely main host or primary host is female Anopheles mosquito. Female Anopheles mosquito is the primary host, and secondary host is man. Generally secondary host mosquito, primary host man, but in case of Plasmodium, based on reproduction, sexual reproduction takes place in the mosquito. Asexual reproduction takes place in the man. So that's why man is intermediate host or secondary host, and secondary host is man. So thus it takes place, life cycle takes place in two hosts, one man and another one mosquito. Each one is 8 marks question. So life cycle in man, that is our heading. Let us turn the page. Life cycle of Plasmodium in man. Compulsory essay question, you know. Out of these two essay questions, one is compulsory. The age-old, stocked, important, very, very important essay question since the long back itself. Since the introduction of this Plasmodium, no exam is there without this question, either mosquito or man. So life cycle of Plasmodium in man, let us see today. This is the beautiful 8 marks question. Very, very important 8 marks question. So you can put number of star marks. It is meant for 4 marks as well as 8 marks and as well as 2 marks. Such a number of questions coming from these two 8 marks. So life cycle of Plasmodium in man. How do you start this question, you know? When the infected mosquito okay, bites, mosquito in the sense which mosquito, you must specify the name of the mosquito also, Anopheles. So when the infected mosquito with infective stages, what are they? Sporozoites. When the infected mosquito Anopheles bites and bites and sucks the blood of man. Sucks the blood of man. The infective stages, sporozoites are injected into the sporozoites, underline. Even in the writing essay question also, answering this type of essay questions also, all important terms should be underlined like this. So sporozoites okay, are injected into the blood of humans, into the blood circulation of human. Okay, into the blood circulation of human. So this is important. So the life cycle in man is divided into, life cycle of Plasmodium in man is divided into 3 stages. In man takes place by asexual reproduction. Asexual reproduction in Plasmodium, what we call, you know, a specific term is there. Already I have given this specific term in the multiple fission. See, in the locomotion and reproduction in protozoa, binary fission, two types of binary fissions we have discussed: longitudinal binary fission, Euglena example; transverse binary fission, paramecium example; and multiple fission also we have discussed, giving examples. So at that time this term I have used. Any one of you recollect? Satvika, you tell me, you answer me today. Satvika. Satvika. Hello. You are there, Satvika? Hello? After reporting, you are going somewhere, huh? You are not there in the class. Bravo? Life cycle in man takes place by asexual reproduction. That is, that asexual reproduction process, one term I have given in the multiple fission. Do you remember? By asexual method called, yes, very good. Called schizogony. In the bracket you write down schizogony. Schizo means split, splitting into number of daughter cells. Schizocoelom, split coelom. Already at the time of coelom also, that schizo meaning I told you. So schizogony, schizo means splitting into number of bits, multiple fission. It's a kind of multiple fission. It is a kind of asexual reproduction called multiple fission, that is so named as schizogony only in case of Plasmodium. Don't write schizogony for other multiple fissions. But in amoebas, multiple fission process is porogony. That's why schizogony you should not use anywhere except in the Plasmodium vivax. Schizogony, life cycle in man takes place by asexual method schizogony. So this is of three types. This is of three types. Occur in liver cells and red blood cells and RBC. Simply red blood cells. Write simply RBC. So liver cells also called hepatic cells. Already hepatocytes I told you. So this is of three types. Occur in liver cells and RBC. Namely, these three types of life cycles are namely: A. Pre-erythrocytic cycle. Pre-erythrocytic schizogony. You can say schizogony. In liver cells, it takes place in liver cells. Exoerythrocytic schizogony. Exoerythrocytic schizogony means outside the RBC, inside the liver cells. That's why second step in the life cycle in man is exoerythrocytic schizogony. Also takes place in liver cells. I am writing here after hyphen where it occurs. So in liver cells, both the first two schizogony occur in liver cells. Last and final one is erythrocytic schizogony or endoerythrocytic schizogony, because within the erythrocytes this occur, RBC. So the schizogony is called endoerythrocytic schizogony, occur in the RBC, liver cells, or the erythrocytes, red blood cells. So like that, life cycle of Plasmodium in man is divided into three stages. The first stage is A. Pre-erythrocytic schizogony, pre-erythrocytic cycle or pre-erythrocytic schizogony. Put a heading like this and let us describe pre-erythrocytic schizogony. Okay, occurs in liver cells. Occurs in liver cells. The sporozoites during mosquito bite. How many? 2000 sporozoites. Write down that point. 2000 sporozoites are injected into the blood. Are injected into the blood through mosquito bite. Through Anopheles mosquito bite. So when the mosquito bites and sucks the blood of man, these are injected into the bloodstream or blood flow or blood circulation. So in the blood they stay for 3 minutes. After 30 minutes are in the blood, thus they stay for only 30 minutes. After 30 minutes they reach the liver cells. And later they reach the liver cells. So like this pre-erythrocytic schizophrenia should be written, and later they reach the liver cells of liver, and feed on the contents of liver, liver contents they feed glycogen, etc., contents of liver cells. And these sporozoites converted into these stages, you must mention, these sporozoites are converted into 3 stages. Converted into trophozoite, a feeding stage, feeds on contents of liver. Schizont, the cell of trophozoite enlarges. That enlarged size is called schizont. And lastly, the cryptomerozoites or cryptozoites, which are 12,000 cryptozoites are produced in the pre-erythrocytic schizont. 1200 cryptozoites. See, very carefully remember. You should underline these stages. Trophozoite, schizont. Schizont divides by schizogony. Schizont, that's why it is so called. It undergoes schizogony to produce 12,000 first generation merozoites and cryptozoites. These are also called first generation merozoites. First generation merozoites. Okay. First generation merozoites, they leave the liver cells and settle in the gaps between liver cells. They leave the liver cells inside the liver only and settle in sinusoids. Sinusoids means spaces between liver cells. In the bracket, if you want the meaning of sinusoids, space between hepatocytes are called the sinusoids. That's why the correct location of cryptozoites, 12,000 cryptozoites, where they are located? They are located in the sinusoids. So like that pre-erythrocytic cycle. So the period of pre-erythrocytic cycle is 8 days. Pre-erythrocytic schizogony completed in 8 days. This numerical status you must remember. Measurement of sporozoite length 15 microns, 1 micron. Number of sporozoites released into the blood by mosquito 2000. So like that. Now how many first generation merozoites are produced? 12,000. So pre-erythrocytic schizogony is completed in 8 days. Within 8 days it is completed. Then what are the end products of pre-erythrocytic schizogony? Cryptozoites. Cryptozoites may again attack liver cells, may again enter liver cells, or may attack the erythrocytes, RBC. This one or that one, either of these can be done by cryptozoites. So cryptozoites are the infective stages to second stage life cycle, that is exoerythrocytic cycle, or third stage life cycle endoerythrocytic. These two cycles are attacked by these cells. But generally the second stage in the life cycle of man, plasmodium in man, is exoerythrocytic cycle. Exoerythrocytic cycle also occurs in liver cells or second hepatocytic schizogeny. This is first hepatocytic schizogeny. This is also occurs in the liver cells. Here I have written, and it also occurs in the liver cells. That's why it is a second hepatocytic schizogeny. So exoerythrocytic cycle initiated by cryptozoites. Initiated by cryptozoites, which are the end products of pre-erythrocytic schizogeny. Cryptozoites also called cryptomerozoites. So it is a lengthy term. That's why we can make it simplify. Cryptozoites occurs in liver cells initiated by cryptozoites. This cycle is initiated by cryptozoites undergo development. Cryptozoites converted into again the same stages in the pre-erythrocytic cycle, converted into trophozoite first stage, schizont second stage, and metacryptozoites third stage, final stages. So these are the three stages, of course not in detail given in the book, but I am giving these stages. Here also and I have underlined trophozoite, schizont, and these end products are trophozoite, schizont common to both pre-erythrocytic and exoerythrocytic. End stages are different. These are cryptozoites, these are metacryptozoites. So these three stages are also there. Cryptozoites are converted into trophozoite, schizont, and schizont always undergo schizogeny to develop into metacryptozoites. Number is not given. Metacryptozoites are generally 1000. 1000 metacryptozoites are developed. So these metacryptozoites, smaller one are micro metacryptozoites. Size varies. Some are small and some are larger. Smaller one are micro metacryptozoites. Micro means small. That's why they are so named as micro metacryptozoites. And larger ones are macro metacryptozoites. Macro means large, micro means small. The meaning itself it is there. That's why you can easily remember. Macro metacryptozoites. So these 1000 metacryptozoites are differentiated according to size variation. Smaller ones are micro metacryptozoites and larger ones are macro metacryptozoites. So exoerythrocytic schizogeny is completed. Second hepatocytic schizogeny is completed. So macro metacryptomerozoites enter the liver cells. Macro metacryptozoites enter the liver cells, but micro metacryptozoites infect the RBC. Micro metacryptozoites start the erythrocytic cycle or enter the RBC or infect the RBC. Anyhow, you can say any way you can speak out. Micro metacryptozoites enter the RBC. So RBC is infected by two stages. Either cryptozoites of, I already have written here, cryptozoites may again enter liver cells or may attack the RBC. Here also I have written that point. So this is important. So two stages. What name the two stages? 2 marks question. Because it is clubbed. This point and this point, these two are clubbed in that question. What is that question? Name the stages which are infective to erythrocytes or RBC. Cryptozoites of pre-erythrocytic cycle, micro metacryptozoites, micro metacryptomerozoites, double underline you should. Micro metacryptozoites here already enter the RBC. These two stages you have to write down. Simple answer. Need not write essays or sentences. You just write those two words. Erythrocytic cycle is started by what are the stages? Cryptozoites and micro metacryptozoites. That's all. 2 marks you will get. So it is completed in 48 hours. 2 days. It is completed in 48 hours. Okay. So here it is completed in how many days? Already I have given here 8 days. These important events should be underlined. So this is 48 hours. So second step, first step pre-erythrocytic cycle is completed. Exoerythrocytic cycle, second step also completed in the life cycle of Plasmodium in man. Very important. Definitely you are sure to get in the IPE, Intermediate Public Examination. This is the last and final one is endoerythrocytic. We start now. Okay. Now let us turn the page. Before starting the endoerythrocytic cycle, this is another 2 marks question, which is what is pre-patent period? That is given. What is pre-patent period? It's also given. So let us see the pre-patent period. The period between the infection of Plasmodium at sporozoite stage, at sporozoite and its first appearance in the blood, that is attacking erythrocytes, and its first appearance in the blood either by cryptozoites or micro metacryptozoites, and their first appearance in the blood is called in the blood, simply in the blood you write down, is called pre-patent period. It is always 8 days. Pre-patent period always 8 days. So many numerical problems are there in the malarial parasite. The period between the infection of Plasmodium at sporozoite stage and its first appearance in the blood, it is called pre-patent period. It is always 8 days. So now either cryptozoites or metacryptozoites start this erythrocytic cycle. So put a heading erythrocytic or endoerythrocytic. Either of these terms you can say. Endoerythrocytic cytogony or cycle of Plasmodium vivax. Exclusively this is 4 marks question. This one exclusively 4 marks question. This pre-patent period is 2 marks question. That is 2 marks, this is 4 marks. Unlike previously, I have discussed, adapted a procedure, that is question bank. I go back to question bank and questions I have used to write on the board and discuss the answers. But at the time of explanation only I am giving which is 2 marks, which is 4 marks and which is 8 marks, like that. So this is in this life cycle again, this is 4 marks question. Erythrocytic cycle or endoerythrocytic cycle discovered by Golgi. Hence Golgi cycle, Camillo Golgi. Our Golgi apparatus who discovered, the same scientist. So discovered by Camillo Golgi, first point in the erythrocytic cycle. So it is also called Golgi cycle. The question may be also asked like that. Describe the Golgi cycle. It is also called Golgi cycle, and the name of the scientist, it is so called as Golgi cycle. Point wise, let us say it is started by either cryptozoites or micro-metacryptozoites. Initiated by or started by. Initiated means started. It is initiated by these stages: cryptozoites or metacryptozoites, micro-metacryptozoites. Micro-metacryptozoites belong to exoerythrocytic cycle. Cryptozoites belong to pre-erythrocytic cycle. So either of these two stages start. So that's why already I told you this is a 2 marks question. At what stages the erythrocytic cycle is started? Cryptozoites, micrometa, cryptozoites, either of these two stages. So now these stages first converted into trophozoite. Trophozoite always a feeding stage. Even Entamoeba histolytica, there also we have discussed that trophozoite. Tropho means food, means feeding stage. Trophozoite always feeding stage. At this stage only they start eating the whatever that is required from the host they are taking. So converted into trophozoite. So then in the trophozoite vacuole appears. In this the vacuole appears pushing the nucleus and cytoplasm towards the periphery. Pushing the nucleus, and diagram I will show everything. Nucleus and cytoplasm towards the periphery. Periphery means around. So cytoplasm towards the periphery. Means margin of the cell, periphery. Nucleus and cytoplasm towards the periphery. So all these stages I will give you the diagram you can see here. So now this stage is called specially appeared stage. Now this stage is like a finger ring, our finger ring, you know. So this ring, how it looks like, same that shape is that. So now this stage is like a finger ring called signet ring stage. Signet ring stage, don't forget, underline. First stage is, what is that? Trophozoite, converted into trophozoite. This is first stage, number 1. Say how many stages are there, correct sequence of stages in the erythrocytic cycle. Also number of times it is a question. So this is signet ring stage. This is second stage. Then vacuole disappears. In this vacuole forms, vacuole disappears. Pseudopodia developed. Vacuole disappears and pseudopodia will be developed. When pseudopodia are there, you can estimate, I can imagine what type of shape that it resembles. Vacuole disappears and pseudopodia appears. Called, looks like amoeba, hence this is amoeboid stage. Amoeboid stage is the third stage. That's why it is called amoeboid stage. This is the third stage in the life cycle of Plasmodium in RBC. First stage trophozoite, second signet ring, third amoeboid stage. Carefully. At the amoeboid stage we have to stop for a while. Why? Because there are so many things happen. In amoeboid stage, all these points we should take at one area. In this amoeboid stage, what events occur, let us see. In amoeboid stage, the RBC enlarges. First event. So many events occur, RBC enlarges. Called hypertrophy. Enlargement of almost RBC double the size. Original size RBC increases to double. So RBC enlarges, called hypertrophy. Enlargement of RBC is called hypertrophy. Already hypertrophy in the parasitic effects of parasitism, hyperplasia, hypertrophy discussed. I discussed. Remember this same word. There also same example we have given. Enlargement of RBC by Plasmodium is called hypertrophy. Double the size of the RBC increases. Hypertrophy, first event. Second event, red colored antigens are released by parasite. Red colored antigens, this is another event, are released, called, most important term, Schaffner's dots. Schaffner is the name of the scientist, so you should say Schaffner's dots. Schaffner's dots. Schaffner's dots underline, most important, 2 marks question. Schaffner's dots again, 2 marks question. This is what are the Schaffner's dots. Antigens, red colored antigens released by the parasite amoeboid stage in the, is called Schaffner's dots. And then third event occurs, the malarial pigment is produced. Malarial pigment, how it is produced, let us see. What is malarial pigment? Malarial pigment called hemozoin. Hemozoin is produced in this stage. Hemozoin formation occurs. It is an orange colored fluid, hemozoin, poisonous fluid. So malarial pigment called hemozoin, poisonous. At the time of liberation of this fluid only, we suffer from chilled fever that appears. So malarial pigment called hemozoin, it feeds on hemoglobin. It eats away, it feeds on third, fourth event. All these occur in the amoeboid stage. So it feeds on globin of hemoglobin. Globin part is eaten away. Globin of hemoglobin eaten away, and heme is converted into hemozoin. Heme part is converted into, heme is converted into hemozoin. Is converted into poisonous fluid. That is called malarial pigment. Hemozoin. So how these events occur, you see, all these events, don't forget to write it. These are all happening in the amoeboid stage. That is the what stage? This is third stage in the life cycle of plasmodium erythrocytes, amoeboid stage. Then the nucleus enlarges, the cell enlarges, cell of the plasmodium enlarges. The cell enlarges by losing pseudopodia. Pseudopodia are lost. So now amoeboid stage becomes round by losing pseudopodia. So when the pseudopodia are lost, it becomes spherical, round. So the cell enlarges by losing pseudopodia, become spherical. Become spherical. Called, this stage is called schizont. This stage is known as schizont. So at this stage, most important event occurs. What is that? You know, the RBC is ruptured. The RBC ruptured in the schizont stage, releasing hemozoin. RBC ruptured in schizont stage and releases hemozoin, a poisonous fluid. So at the time of liberation of hemozoin, the malarial fever symptoms appear. Malarial fever symptoms appear, hemozoin. So during which the fever symptoms appear. Details we can give later on. Just one word you add here, one phrase. During which the fever symptoms appear. During the release of hemozoin, the fever symptoms appear. So at what time the fever symptoms appear? At the time of liberation of hemozoin by the rupturing of RBC during schizont stage, hemozoin is released. So that is. And then schizont undergo erythrocytic schizogony. Schizont produces daughter cells 12 to 24. Schizont produces 12 to 24. This number also remember. Daughter cells. called merozoites. So this is the fifth stage called merozoites. 12 to 24. They are irregularly arranged. Life cycle almost completed. Which are diagram tomorrow we can draw because already time. Which are irregularly arranged. Sometimes they are regularly arranged like a rosette, rose flower like. So which are irregularly arranged. Last one heading is incubation period. This is also 2 marks question, like pre-patent period. Incubation period. What is incubation period? Let us define. A period between the entry of parasite, entry of Plasmodium, and first appearance of symptoms. Entry of Plasmodium at sporozoite. You need not write down sporozoite. Entry of Plasmodium. And first appearance of symptoms is called fever symptoms. First appearance of fever symptoms known as incubation period. Known as, I need not write known as. Heading itself. It is 10 to 14 days. Incubation period is 10 to 14 days. 10 to 14 days is the incubation period of this parasite. 8 days is the pre-patent period of the parasite. Here already I have written. So these two are 2 marks questions. This is also 2 marks question. See how many 2 marks questions are there and 4 marks questions are there in this essay question. Essay question is completed. Tomorrow I draw the diagram of all these stages. So describe the life cycle of Plasmodium in man with the help of diagram. It is asked always. Okay, I can stop the class. Thank you. Okay, ma. Okay, bye.